Hello, everyone, and good evening. This presentation is on the topic of the Human Genome Project. Introduction to the Human Genome Project, or HGP. The HGP was an international scientific collaboration to completely sequence the human genome and to identify all the bases that make up human DNA and to identify and map all the genes of the genome, both from a physical and a functional standpoint. The project began in 1990 and was declared complete on April 14th, 2003, 2 years ahead of schedule and at a cost lesser than the original estimated budget. It is well known that the human genome shows 99.9% similarity within the species. However, it is the 0.1% of variation that gives such diversity in the population. Hence, with the samples obtained from a small number of individuals, sequencing doesn't provide a map for every individual in the species. Rather, a mosaic that doesn't represent a single individual, but a species as a whole, with variation consideration within the sample size. The timeline of the HGP. In 1998, the NIH director assembled experts and scientists to plan out the HGP, and in 1989, the Office for Human Genome Research was established. In 1990, a 5-year plan was laid out and the official goals are published as the HGP officially begins. In 1994, the first mapping goal was met and the linkage was published a year ahead of schedule. In 1996, the NIH announced a pilot study to study large-scale sequencing of human DNA. In 1998, some projects were extended an extra third year for refinement, and the plan to finish 2 years ahead of schedule is released. By 1999, the pilot phase was completed, and the sequencing of 3 billion bases of human DNA is launched, and leaders of the project guarantee free access to any information obtained from this project, and the first draft was scheduled to be released by the year 2000. In 2000, 2 billion of the 3 billion bases have been deciphered, and the first draft of the Human Genome Project is assembled. By 2003, the HGP was completed 2 years early, and the NHGRI selected 5 centers to continue further large-scale projects associated to the HGP. The goals of HGP. The first was genetic mapping, obtaining a complete map of the human genome and to develop technology for rapid genotyping. The second was physical mapping, a complete sequence tag site map or an STS map of the genome. Third was DNA sequencing, development of technology for high throughput sequencing. Fourth was gene identification. 5. Technology development. 6. Sequencing of model organisms like E. coli, yeast, Drosophila melanogaster, and mice genome. 7 was informatics, development of more databases and tools and platforms for data exchange, comparison, and interpretation of information, and easy access to data has to be provided. 8 was ELSI, the ethical, legal, and social implications of the Human Genome Project. 9 was training for scientists in the interdisciplinary sciences that are related to human genome research. 10 was technology transfer, and 11 was outreach. Coming to the people who participated in the Human Genome Project. It was very well known that the Human Genome Project was an international collaborative effort. So there were many institutions and universities that were involved in this, such as the Whitehead University at MIT, the Wellcome Trust Sanger Institute, Washington University, the University of Oklahoma, Baylor College, Stanford University, the Beijing Genomics Institute at the Chinese Academy of Scientists, the University of Texas, the University of Washington Genome Center, the Max Planck Institute for Molecular Genetics, the Department of Molecular Biology at Keio University School of Medicine, etc. Coming to the applications of Human Genome Project. The sequencing of the human genome provides a lot of information that can hold many benefits in many fields. Fields that involve genotyping of specific viruses to provide direct appropriate treatment, to identify mutations linked to different forms of cancer, to design medication and more accurate prediction of their efforts, advancement in forensic and applied sciences, biofuels and energy applications, agriculture, animal husbandry, waste control, environmental cleanup, risk assessment, bioprocessing, bioarchaeology, and anthropology and evolution. The sequence of DNA was also made available on the NCBI GenBank, and the usage of programs to analyze the large amount of data obtained was seen. The usage of tools to speed up the sequencing process was involved. There was also a requirement to understand 0.1% of difference or variation in the genome that could lead to discoveries in healthcare and advancements in many treatments of diseases such as Duchenne's muscular dystrophy, retinoblastoma, cystic fibrosis, neurofibromatosis, different types of cancers, Alzheimer's, and Fragile X syndrome. Coming to the case studies related to the Human Genome Project, which are the implications of the Human Genome Project for medical science. Now, only recently has the implication that heredity in a disease is not just for guidance and medical surveillance, but it is being moved beyond that to the idea to establish its role in the molecular basis for these diseases and their influences in this disease progression. Moving to the current research focus with the implications of the project in medication. Additional work needs to be done to completely sequence the entire human genome and complete sequence analysis by closing gaps and resolving ambiguities. The sequencing for other organisms also needs to be done so that we can establish evolutionary relationships between them and humans. It is also required to develop a catalog of human variants to give clues about genetic diseases or their risks in illnesses. Different technologies and methodologies to study gene expression need to be developed. And an emerging field of computational biology needs to be seen so that we can gain insights from the vast amount of information that we have seen from the project. We also need to draw awareness towards the ethical, legal, and social implications of these research programs. Coming to the 21st century and the critical elements of medical research agenda. Information about the genome needs to be used in the application and identification of genes that play a role in the hereditary contribution to diseases and to identify options for combination of phenotyping and sampling of genetic variants across the genome to identify gene associations in the diseases that is seen in diseases such as diabetes mellitus, where various different genes with various different magnitudes of risk are seen to interact together to create the symptoms for the disease. Next, methods need to be developed to design drugs and to modulate disease pathways and alter them in ways that can change their direction into something that we desire. We need to involve methods that systematically combine chemical components into drugs and of high volume assays for efficacy and to identify, stimulate, or block a particular pathway, like seen in the drug STI 571, which was designed to treat the disease of chronic myelogenous leukemia. Genomics also provides opportunities to test for drug responsiveness and the variation of these drugs in response to the genetic endowment of a specific individual. It is also required in the safer use of gene therapy and applications of gene therapy for different diseases. Coming to the genetics in the medical mainstream, it is expected that predictive genetics tests will be available to common citizens to treat dozens of common conditions, and it has been given to individuals who want to know their genetic endowments and their individual susceptibilities so that they can take steps to reduce the risks from these illnesses. Another crucial step is to pass effective legislation to outlaw the use of predictive genetic information in the workplace and in obtaining health insurance, and to ensure that there is no discrimination based on genetic information if required. Next is the requirement of new gene-based designer drugs, which when introduced to the market can be helping in treatment of diabetes mellitus, hypertension, mental illness, and many other conditions. With these new and improved treatments of diseases like cancer, it is most likely that the clinical sector and the clinical industry will be majorly advanced with the consequences that are seen in genetic studies. To conclude, I would say that the efforts with the Human Genome Project are to provide many wide avenues in many fields that are not just tied to genomics, but in the medical industry, in the environmental fields as well, just to name a few. With the information that is obtained, we can use it to provide many benefits and thus to hopefully answer the big questions that can help us evolve and advance together as a species. Thank you.